Proteins are hydrophilic polymers that are made up of amino acid monomers. When you think of a protein, what food comes to mind? Probably animal meat, but those are made up of proteins and also fats and carbs. You can also get a lot of protein from plants. Things such as beans, legumes, and other whole wheats are full of proteins. The monomers of proteins are called amino acids. There are 20 different amino acids and infinitely many ways to build a protein. This means that they can have many different structures, which means they can have many different functions. One of the most important functions of proteins are enzymes. These are catalysts that speed up reactions in our bodies. Without enzymes, we wouldn't be able to process food or make energy quickly enough to meet all of our needs. Proteins can also be involved in transport. Many proteins in our blood carry molecules throughout the body. Oxygen binds to the protein hemoglobin. Some proteins are signal molecules, called hormones, that communicate with other places in the body. Proteins can even be used in defense. The antibodies we use for fighting off pathogens are made of proteins. Muscles are full of contractile proteins, such as actin and myosin, that result in the movement of muscles. We use proteins for storage, such as those found in egg yolks or seeds. The unique function of each protein depends on its unique shape. If the shape of a protein is disrupted, it can no longer function. This is called denaturation. When a protein is denatured, it cannot be fixed and is permanently broken. This is caused by high temperatures, pH, and other environmental factors. This is what we do when we cook food. We denature the proteins in potential bacteria cells so that they are not functional and can't hurt us. Amino acids are the building blocks of proteins. There are 20 different amino acids. Each one has the same basic structure. Look at the amino acid structure in the top right. We'll start with the center carbon. All of them have 4 things attached to the central carbon. 3 of these are the same in all 20 amino acids. On the left of the carbon is an amino group, NH2. And to the right is a carboxyl group, C double bonded to O, bonded to OH. All amino acids have these structures, in addition to the hydrogen above the carbon. The difference between the 20 amino acids is the R group. The R stands for a variable, kind of like how you would put X in an equation in algebra. The R has 20 different possibilities, making up the 20 different kinds of amino acids. The function of each amino acid depends on its R group. Some are polar, some are nonpolar, some are acidic, and some are basic. When 2 amino acids are linked together, they form a covalent bond between them called a peptide bond. When a lot of amino acids are linked together into a long chain, they form a polymer, which is called a polypeptide. A long chain of amino acids doesn't function like a protein yet. It has to be folded into the proper shape before we call it a protein. Proteins can be broken down into 4 levels of structure. The lowest level of structure is the sequence of the amino acids. The order that the amino acids go in predicts the ultimate structure of the proteins. Think of the sequence like words in a sentence. If you change the order of the words in a sentence, it doesn't mean the same thing anymore. Same thing works here. If you change the order of amino acids, they don't make the same protein anymore. The secondary structure is coils or folds. These are associations between adjacent amino acids based on how their hydrogen bonds form and interact. The tertiary or third level of structure is the overall 3-dimensional structure of the polypeptide. This happens when the coils and folds are folded back up on each other to form a 3D molecule. Some proteins are fully functional at this point. But many proteins have more than one polypeptide chain. The quaternary structure is the final association of several different 3D structures of multiple chains. These larger proteins, like hemoglobin, have 4 polypeptide chains, and this makes up their quaternary structure.